Hello student, this is Dr. Vaibhav Pandit. In this video we will discuss about the functions and deficiency symptoms of the micronutrients, essential micronutrients. First one, that one we are going to discuss, that is iron. So plant will take the iron in Fe2 plus form, and this sufficient range in a plant is 100 to 500 ppm. If the iron content is less than 100 ppm, then plants will show the deficiency symptoms. Then iron is a one transitional metal, so it means that it will present in two forms, that is Fe2 and Fe3 forms. So because it is a transitional metals, it will always take up parts in the redox reactions. So here we will see the some functions of the iron in the plants. So plants play the major role in the nucleic acid metabolism, like the DNA and RNA formation. It's it plays a major role in the chlorophyll formation and maintenance. So iron is also the part of different types of proteins. So there are two groups of protein. One is the heme proteins, another Fe and S proteins. So in heme proteins, it is a part of the cytochrome oxidase, catalase, and peroxidase. This is responsible for the formation of porphyrin complexes, which is responsible for the chlorophyll formation. Then FES protein, which is uh, responsible for the or a part of the ferredoxin. So ferredoxin is formed by the Fe and S. So that is responsible for the reduction of the different oxidizing compounds, like for example reduction of the nitrate into the NH4, like that. So if you see here the some functions through the some good diagram. This is the mitochondria. If you see the respiration here, the cytochrome oxidase enzyme, which is activated by the iron. So iron is a part of cytochrome oxidase, followed by the FES protein, that is lake hemoglobin proteins. So lake hemoglobin proteins, which is responsible for the biological nitrogen fixation. So is the lake hemoglobin. Fe is a part of lake hemoglobin. Then this is the FES protein, that is non-heme proteins. So it is also the part of pyroxene. Is iron is also part of the pyridoxine. Then it will activate different enzymes which is responsible for the formation of chlorophyll. The enzyme is like uh, amino leucinic acid synthesis and coproporphyrin oxidase. This enzyme is responsible for porphyrin ring synthesis. If the iron is not there, these two enzymes will not form, then chlorophyll will not develop. So we will see the yellowing of plants. Here we see the complete ALAS enzyme activation by the irons and converted into the porphyrin ring. Also this one coproporphyrinose oxidase enzymes, which is acted by irons, and these two enzymes responsible for the chlorophyll formation. Then coming to the deficiency symptoms of iron. So mostly the iron deficiency will occur in a plant when the iron content in the plant is less than 50 ppm. Okay, so first symptoms of iron will appear on the younger leaves, because iron is a immobile elements. That's why the deficiency will comes on the younger leaves. So two types of the elements will be there in a plant. One is the mobile and another one immobile. So mobile elements like nitrogen, phosphorus, potassium, magnesium, these are the immobile elements. So their deficiency mostly occur on the older leaves, whereas the immobile elements such as iron, here the deficiency will occur on the younger leaves. So deficiency symptoms of the iron is the intervenous chlorosis. If you see here the plant, these are the intervenous chlorosis. Here this is the intervenous chlorosis. Okay, so vein and margin will be green, the internal part will become the yellow. So vein is green, margin is green, the internal parts become yellow. That is called the intervenous chlorosis. So it will often in different crops like maize, citrus, and tomato. This is common. If you see the normal crop here, this is green, normal crop is green. Then if the iron deficiency is there, it is the yellowing of top portion, then stunting of growth, then necrosis is becoming the brownish in color, and then death of the plant. So these are some different symptoms mostly occur in the plant. Then coming to the another element, that is manganese. Manganese plant will obtain in a Fe2 plus form, where the sufficient range of manganese will be between the 20 to 30 ppm. Manganese also the transitional metal, so it means that it will present in three forms: Fe2, Fe3, and Fe4 forms. So because the transitional metal, it will take a part in the redox reaction. Mostly, it's play the major role in the photosynthesis and respiration reactions. We'll coming to the function of manganese. Manganese play the major role in the photosynthesis, especially in the PS2 cycle by splitting the water molecules and the evolution of the oxygen from the photosynthesis that will occur through the manganese. Then manganese is a part of the SOD enzyme, that is superoxide diamidate enzyme, which that enzyme which protect the superoxide free radicals like Fe3, which damage the cell. So if that enzyme is there, will capture the superoxide free radicals and avoid the damaging of cell. Then manganese also play the major role in a TCA cycle, that is Krebs cycle, and activate the different enzymes which relate to NDA. Then it's also play the major role in the carbohydrate and nitrogen metabolism. We'll see here in a diagram. This is the water splitting mechanism in a pH2 cycle, and this is here the Krebs cycle, how manganese is responsible for the decarboxylation. And another function of the manganese is the defense function, that is superoxide dismutase enzyme activation. If the cell is unprotected from the superoxides and is stressed, then damage will damage. Can you see here? This is the damaging of cell. This is superoxide, that is it will damage. It will damage the cell. It will damage the cell. But the Mn is present, then Mn will release the SOD, will activate the SOD and protect the cell from the superoxidase. Then also it will take a part in carbohydrate metabolism, like the conversion of the carbohydrate. Also take part in a glycolysis and respiration, and also it take part in nitrogen metabolism. Coming to the deficiency symptoms of the manganese, manganese and iron almost are the mobile in the plants. So it is a mobile element, so the deficiency symptoms will occur in the younger leaves. So here also we will see the intravenous chlorosis, but it is different from the iron intravenous chlorosis. Here in a intravenous chlorosis, so after intravenous chlorosis, here due to manganese intravenous chlorosis will occur. After intravenous chlorosis, there is the appearance of the brown spots. Then brown spot is converted in necrotic area. This we can see the complete brown spot. In oat, there is a gray speck disease due to the manganese deficiency will occur. The symptoms is the like intravenous chlorosis followed by the brown spot on the lips. This is the deficiency symptoms of manganese. Then another micronutrient, that is a zinc. The major role of the zinc is the synthesis of oxygen, like IAA. Also, it is responsible for gibberellic acid metabolism and the RNA synthesis. So it is indirectly take a parts in a protein synthesis by binding the SH group. This group is responsible for the formation of proteins. Then under if you see the function, major function is activate the different enzyme like carbonic anhydrase, alcoholic dehydrogenase, and superoxide dismutase. Carbonic anhydrase is required for the CO2 fixation for the photosynthesis cycle. Then alcoholic dehydrogen required for the anaerobic respiration, and superoxide diamide, we know that they is responsible for detoxification of free superoxide radicals. Here we'll see here the function of zinc, that is the fixation of the CO2 by activation of enzyme, that is carbonic anhydrase. Then the activation, another example, then alcoholic dehydrogenase, which is responsible for anaerobic respiration. Then another third important enzyme, that is superoxide dismutase, which is required to protect the cell from the damage by the superoxide. This is the some relationship between the zinc and phosphorus. If the phosphorus is more in a plant, the zinc deficiency will occur in a plant. Both are negatively related to each other. So deficiency in terms of zinc, the zinc deficiency will occur in the plant if the zinc present in a plant less than 15 ppm. Here also we'll see the intervenous chlorosis in young leaves. It means that the zinc is a mobile element, immobile element. Sorry, it's the immobile element. Then there is a due to deficiency there is a reduction of the lip size, then converted into browning and followed by the reddish and brown discoloration, you know, on the leaves. Then if you see the dicot plant, we will see the rosety. Rosety is the uh, short internode distance. Then the reduction of the leaf area, that is called little leaf. So some of the most well-known diseases we'll see in a rise that due to the deficiency of zinc, that is Khaira disease, which is occur due to the deficiency of zinc. In maize, we'll see the white bud or mottle leaf disease due to the deficiency of zinc. In citrus, that is little leaf. In cotton, also that is little leaf disease. This see if the zinc is normal, you will see the plant is healthy. There is no deficiency. It is green color. If that less than 15 ppm zinc, there is a deficiency. You will see the intervenous chlorosis. Young plant will get affected. If the more deficiency is there, less than 10 ppm, then it is turned into the brownish color. Then there is a necrotic spot will form. This is a healthy dicot. There is no deficiency of zinc. Here the Khaira disease, where the plant, green plant, this is a rice plant, converted into completely brown in color. That is also known as the uh, missing hill area. This is also known as missing hill area. This is a deficiency of this is a reduction of the plant leaves due to the deficiency of zinc, that is called little leaf. The size of leaves is small due to deficiency of zinc, that is mostly found in the cotton and citrus. This is a white bud in maize due to deficiency of zinc. In cotton, little leaf area in cotton. Then another micronutrient, that is the copper. Copper will uptake by the plant in CO2 plus form. The sufficient range of the plant, sufficient range of copper in a plant, that is between 5 to 30 ppm. So it is a so there is a function of copper. Copper is a part of many enzymes like flastocyanin, superoxide dimutase, diamine oxidase, polyphenol oxidase, ascorbate oxidase. So flastocyanin is a part of the photosynthesis. Is responsible for chlorophyll formation. Superoxide dimutase is responsible for uh, detoxification of free radical. Diamine oxide responsible for protein formation. Polyphenol formation is responsible for lignin formation. Ascorbate oxide responsible, or it will activate when the stress is there, more stress, and ascorbic acid will form. So to counter that ascorbic acid, plant will release the ascorbic oxidase to avoid the leaf shading. So the copper is there, ascorbic oxide will produce and leaf shading will reduce. So copper will play the major role, that is a dis resistance in a plant due to the increase in the strength in the stem. Then it enhances the male sterility due to increase in the uh, phospholignin contained in the plant. We'll see here. So it is a part of different enzyme like ascorbate oxidase, polyphenol oxidase, diphenyl oxidase. Then phastocyanin, which plays a major role in a PS2 cycle. In SOD, it is used for scavenging of radioactive oxygen species. Then it is used for lignification by activating the polyphenol oxidase enzyme, that is responsible for lignification and cell wall strengthening. Then it will that lignification is very important. It will play a major role against the pathogen. That's why we will say the copper is present in the plant. The lignification is more. The cell wall strengthening will be more. and it will work against the pathogen. So the disease resistance symptoms in the plant, disease resistance function in the plant, that is because of increasing the lignification and cell wall strengthening. Then under the moisture stress condition, there is one hormone we release, that is ascorbic acid. To counter that ascorbic acid as well as leaf shedding, the plant will release the ascorbic oxidase. For that, copper is required. If the plant will release the ascorbic oxidase, then there is a no shedding, and there will be the conversion of the ascorbic acid into another compound. So that will avoid the shedding of the leaves. Then copper will improve the male sterility in the plant and also increase the pollen sterility. Then the deficiency symptoms of the copper. The copper deficiency symptoms will occur in a plant when the copper content is less than 5 ppm. So most of the problem with the copper deficiency is the male sterility, then delaying of the flower and shedding of leaves. If the shedding of leaves called senescence, that will occur due to the release of the ascorbic acid in a plant due to the absence of copper. Delay in the flowering because there is a deficiency of copper. Then the visible symptoms is like the chlorosis of Engelus because it is the immobile element. Then white tip, then reclamation disease. Reclamation disease is nothing but the plant will turn into complete white in color, so that's why it's called reclamation disease. Followed by necrosis, leaf distortion, and dieback. These are the some visible symptoms we'll see in a plant. So deficiency mostly occur due to the copper content is less than 5 ppm. Male sterility will occur, delay in the flowering, then senescence. This is the aging of plant, that is called uh, shedding of plants. Then intervenochlorosis, then white tip, then dieback distortion of leaves, then necrosis. This is the reclamation disease due to the copper, that is the complete turning of the green plant into the white in color. There is a dying of the shoot, so we can say that is the stopping of the growth. Dying of shoot mostly occur in the angules because the copper is the immobile in plant. Then next one is the molybdenum. So plant will take the molybdenum MnO4 minus 2 form. The sufficient amount of iron required, a molybdenum required for the plant, that is 0.1 to 2 ppm molybdenum. So it will exist in different form, that is molybdenum 4, molybdenum 5, and molybdenum 6. But plant on molybdenum 4. Then molybdenum is a part of different enzyme like nitrate reductase. That nitrate reductase required for the reduction of uh, nitrate into the NO2, then NO2 to NH4, and that nitrate reductase present in the cytoplasm. Then it is a part of nitrogenase that is responsible for biological nitrogen fixation. Then also it is a part of, or it will activate the xanthine oxidase at the hydrogenase enzymes. It also activate sulfide oxidase. Then molybdenum is a part of nitrogenous enzymes that is responsible for biological nitrogen fixation, which convert NO2 to NH3. It will also help in protein formation. Then it will involve in the nitrate metabolism, that is like the by activity in the nitrate reductase. It will take parts in the nitrate metabolism. So it plays a very essential role like pollen formation and development of anther. Without molybdenum, the pollen formation and development of anther is incomplete. So if the deficiency is there, we will see the pollen formation or the flowering will not be good. If the flowering will be good, there is a no fruiting to the plant. Then the deficiency will occur when the molybdenum less than 0.1 ppm. The deficiency will always look like a nitrogen because the molybdenum is also the mobile element. So its deficiency will mostly occur on the older leaves. So requirement of molybdenum to the plant is very less compared to the other essential elements. Total essential elements are 17. If you see those 17 elements, and if you see the sufficient amount required for the plant in case of molybdenum, the molybdenum requirement is very less. That's why this molybdenum is also called ultra micronutrient element. So if you see the visible symptoms of the molybdenum, which will occur on older leaves because it is a mobile element, there is a mottling between the veins, chlorosis, then mottling in the veins, stunted growth, and distortion of the leaf. That is the most common distortion of the leaf. Then, if you see in cauliflower, there will observe the whiptail disease due to the deficiency of molybdenum. Here we will see the malformed leaves, that is the distorted leaves, narrow leaves. In brassica crops, we'll see the chlorosis, then cupping of leaves. If there is a deficiency of molybdenum in a plant, then we will see the poor nodulation, less nitrogen fixation, less conversion of the nitrate in NH4 in a cytoplasma, then a less formation of the protein. These are the deficiency we'll see. Then the quality of the grain will reduce. Then here we'll see some deficiency symptoms of molybdenum. So here you see uh, the uptake of nitrogen in the plant, or we can say the use of nitrogen by the plant, it depends on the molybdenum. It depends on the molybdenum. So here we'll see the nitrate enzyme which is activated by the molybdenum, which is responsible for conversion of nitrate into the NO2, then NO2 to NH4, then it will convert the amino acid and protein. So for that molybdenum is required. If the plant is healthy, we'll see. We'll see the um, chloritic mottling here the between the veins, between the then deformation of leaves, deformed leaves. Here we'll see in the cauliflower that is whiptail disease, that is the narrow and the deformed malformed leaves. In brassica, that is cupping of leaves, and intervena chlorosis, that is common symptoms in cabbage. Common symptoms in a cabbage. And we'll see in a legume there is a poor nodulation. Poor nodulation reducing the biological nitrogen fixation. Here the difference between the healthy plant due to the high or sufficient amount of molybdenum. This is the deficiency of molybdenum in the cabbage. You see here the cupping of leaves or malformed leaves. Is a severe deficiency molybdenum. Then if you see the root comparison between molybdenum sufficient plant and molybdenum deficient plants, the stunted growth will form in the roots. Here the nodule quantity will reduce in a molybdenum deficient plant. Then coming to the next element, that is the boron. So boron is a anion. So plant will take the boron in the form of H3BO3 form. Also it is present in a, it will also plant will take another form, that is H2BO3, HBO3 by 2 minus 2 and BO3. Under the high pH condition, this form will be available. That's mostly these two form. Slightly acidic condition, this form will be there. So this form also will be available. Boron is available to the plant between 6.5 to up to 8 pH. After that, there is a deficiency of boron in a plant. The function of boron is the formation of cell wall. Formation of cell wall. Then is also responsible for the cell wall elongation and the cell division. Like molybdenum, it plays the major role for the boron germination and boron deformation. It is responsible for ATPase enzyme formation and also the transformation of H plus ion between the cells. It will maintain the potassium quantity in the guard cells as well as calcium also in cytoplasma. Here we see the cell wall pectin or polysaccharide are the linkage between the cell wall pectin. The requirement for them, the requirement is boron. So if the boron deficient sufficient in the cell wall, will seal the cell wall this type. The deficiency is there, will seal the broken cell wall. Here the boron sufficient is there, the cell wall division is good. Here the cell division is not proper. So the stunted growth we can see. Cell elongation same. Here the sufficient boron, cell elongation more. Here the deficient boron, cell elongation is less. If you see here, the pollen germination is good in the boron sufficient plant, and pollen germination is reduced as compared to the boron deficient plant is compared to boron sufficient because pollen tube formation will not occur properly in boron deficient plant. Then for iron uptake, there is a need of H plus ATPs. That H plus ATPs quantity which is present in the plasma membrane. So that one is depends on the also in boron. That boron will decide the H plus ATPs which is responsible for the uptake of potassium, iron, and other elements. Then next one is the stomata opening and closing, which is depends on the potassium. But the concentration of potassium in a guard cell, which is also controlled by the boron. Then what is the different symptoms of the boron? Here we'll see like the hollow stem in the different crops, especially in the cauliflower. Then the death of the growing tip, like apical meristem, because boron is a immovable element, so is the deficiency mostly on the tip portion of the plant leaves. Then due to the deficiency of boron, there is a deformation of leaves, thick leaves we will see, the brittle leaves. The fruit setting flowering will not be good because the deficiency of boron, it will affect the pollen deformation. Then cracking and corking, corkiness in the fruits and stem, that is the major symptoms we will see. This is the hollow stem problem in cabbage. We'll see this hollow stem problem in cabbage. This is the blackened tip and dying, that is death of the growing tip because of the deficiency of boron. Brittle, thick, and deformed leaves in cabbage. Cracking in tomatoes, even in apples, in sugarcane, many stems. This is withering of the leaves, then the poor flowering and fruit setting. Cockling in the stem, inside the stem, this cockling like feature we'll see. So boron also plays a major role for the fruit setting and flowering. Then another one element is the nickel. So we'll see the function of nickel. Nickel is associated with the nitrogen metabolism, so it will activate urease enzymes. So nickel is a recently added element in the list of essentiality. So it is given by the Brown. So if we'll add the urea and or urea spray will give, if the poor nickel supply is there, the urease activity will not occur, so we'll face the urease urotoxicity, then necrosis and tip burn. So also nickel is supply is also responsible for hydrogenous activity because rhizobium nitrogen fixing bacteria to fix the biological nitrogen fixation, hydrogenous activity should be there. So that hydrogenous is depends on the nickel. Then it will transfer the nutrient to the seeds or the grain. So finally, if you see the function of the nickel, one is the activity of urease or the metabolism of nitrogen or urea. Second one is the it will help in the biological nitrogen fixation by active the nitrogen activity. That nitrogenous enzyme help for the nitrogenous another enzyme which is activated during the biological nitrogen fixation. Then it will facilitate the nutrient movement to the seeds and grain. You'll see here, this is the healthy plant where the nickel supply is high, urea is there, urease enzyme is there, so there is no toxicity. Here the nickel supply is there. If you spray the urea will be there, urease will not form. There will see the toxicity of urea. That is then browning will occur, necrosis will occur. Then the nitrogen enzyme required for biological nitrogen fixation. We can see here in figure. If the no nickel is there, then we will not get a correct output. And if you see the deficiency will of nickel occur if the in nickel content in the plant less than 0.1 ppm. So if the nickel deficiency is there, there is a reduction of the dry matter weight, then decrease in the amino acid content and the accumulation nitrate, then reduction of the protein content in the plant, and because of due to reduction in the biological nitrogen fixation. The deficiency symptoms we will not see in India. So then another element that one we like to see, that is uh, chlorine. Another element that one we like to see, that is chlorine. So chlorine, plant, it is a anion. Plant will take the chlorine in a Cl minus form. So like manganese, chlorine also plays a major role in a pH2 cycle by splitting the water molecules. That is a major role. So if you see, the major role of the chlorine is the use as a disease resistant element or pest resistant element. If the deficiency, if we see the chlorine, there is deficiency of chlorine is the formation of brown spot on leaves. Is the formation of brown spot. Yes. If you see the deficiency of chlorine, there is a formation of, there is a development of this rice sheet belt. The deficiency of chlorine conversion. This is intravenous chlorosis. This is the intravenous chlorosis. Then the attack of fusarium in the absence of the chlorine. If the chlorine content is less, fusarium attack will be more. So that is one of the sin. Then powder mildew attack also will be more in different crops. So if the chlorine content is more, so what is the maximum disease we can control, like powder mildew, downy mildew, fusarium, any fungal diseases we can control. So we'll stop here. We will see the nutrient movement in next lecture. Thank you.